Okay, it looks like a lot of our participants have been able to join us. Good morning, everyone. I'm so thrilled that you could be here with us this morning for this important briefing on COVID-19, its variants and sequencing. I'm, as some of you know, State Senator Christine Cohen, representing the 12th Senate District in Connecticut, and one of the Bioscience Caucus co-chairs here in the Connecticut General Assembly. Joining me today are a couple of the other co-chairs. I don't believe Representative Steinberg is with us yet this morning. Hopefully he can break away and join us. I know we all have incredibly busy schedules. And we've got Senator Tony Huang and Representative David Yaccarino, all my fellow co-chairs. I'm just going to kick it over to Senator Huang for a moment to give a brief introduction of himself, and then we'll go to Representative Yaccarino. Tony?
Thank you, Senator Cohen, and thank you so much to Jackson Labs and Yale and all the researchers out there that are making a huge difference, positive difference in people's lives in Connecticut and throughout the country and world. And I am indeed State Senator Tony Huang, and I'm so proud and honored to be part of this bipartisan Bioscience Caucus. It is an important and integral part of our business and quality of life standards in Connecticut. So thank you for joining us, and I'm honored to be here. And thank you to Senator Cohen for helping coordinate this effort as well.
Thank you, Senator. Representative Yaccarino.
Thank you, and good morning to all. Thank you, Senator Cohen and Tony Huang, Senator Huang, and Jonathan Steinberg was not here this morning. I just want to give a great thanks to Jackson Labs, to Yale, and all the researchers, the scientists, and people that work in the industry to help combat COVID-19 and all the other viruses or any other, just anything to do with science, bioscience. I'm proud to be part of this caucus and to work for this initiative over the last 6 or 7 years, and I'm eager to listen. I know the variant is evolving like any virus does, and thank you for all for being here and to giving us an update this morning. Thank you. And thank you to Lisa Roy also for organizing this with Senator Cohen.
Thank you. Representative, yeah, it's so important for us as legislators who are hearing from our constituents all the time about what is happening out there with this virus. And so we're so appreciative that you've been able to take the time today to debrief us. I know that many of our colleagues registered to hear this presentation today from the Jackson Laboratory and the Yale School of Public Health. I know we have Dr. Mark Adams of Jackson Labs here today, as well as Dr. Nathan GruBaugh of Yale, and Lisa Roy of Jackson will be our moderator. We will have time for question and answer after the presentation. So again, I think you heard use the Q&A function of Zoom down at the bottom to type in any of your questions. So we're just so grateful to have these amazing scientists and their facilities on the front lines in this research and testing, so we can learn more about COVID, this virus, and as I said, its variants and sequencing. So just really grateful for all that you've done and all that you continue to do to work to combat this virus and learn more. And so with that, I'll turn it over. Is it Dr. GruBaugh that's going first, or Dr. Adams?
I'll be going first. It's Mark.
Oh, thank you. Thank you. Welcome, yeah. Dr. Adams. Please feel free to tell us a little bit more about yourself and your work, and look forward to hearing your presentation.
All right. Thank you very much for the opportunity to present, and again to Lisa for organizing, and Nate's going to give a great summary of what's going on in the state right now. I'm a professor and deputy director at Jack's Genomic Medicine here in Farmington, and have been working with the CLIA lab off and on over the course of the last year as they've scaled up dramatically for doing SARS-CoV-2 testing in the state. And so starting back in mid-March, we began doing qPCR testing for SARS-CoV-2. And while that was going on, over the course of all last year, our answers were detected and not detected. That's all we knew about a sample. But against that background, there were many different variants, lineages, strains, varieties of SARS-CoV-2 that were circulating throughout the United States, as shown in this graph, that were characterized by different genetic changes that were happening really from the very beginning as the virus evolved and spread throughout the world. And in fact, some of those early mutations probably contributed to more rapid spread as the virus left China and moved to other parts of the world and adapted to those local environments. Starting back in late December, mid-December, as a result of an extensive sequencing program in the United Kingdom, there started to be word of this new type of virus that's now called the UK lineage. It's called 501Y.V1. It's called B117, but those are all synonyms. But the rate at which that was increasing really caused concern. And the identification by sequencing, given the intensive program in the UK, really gave an early warning of that, the emergence of that strain. So I've used all these words now, variant, lineage, strain, and it sounds like I'm kind of using them interchangeably. And in fact, that's a fair enough thing to do. These words come from different scientific fields, whether that's public health, evolutionary biology, or microbiology. And to a good approximation, they are synonyms for one another. And so I would say it's not inappropriate to use any of them depending on which your preference is. And if you want to be more specific, that's okay too. But what they share is a set of mutations that define the variant or the lineage. And those mutations are shared by strains that have a common ancestor, evolved together. And those mutations can in turn affect the function of the virus, either how it's transmitted, how it responds to the immune system, how it responds to therapy, or other things, how severe the disease is that it causes. And it can take very few changes in a virus to have these effects, which is why both the precision of genomic surveillance is important, as well as tracking that over time, because it just doesn't take very much to cause these differences. The reason is that the SARS-CoV-2 genome is actually relatively small. It's about 30,000 bases long, of course, much, 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 much smaller than the human genome, and it only encodes 29 proteins. But there's no junk in this genome. It's very information dense, and all of those base pairs have some important role to play in one of the various factors in the viral life cycle. And so even one mutation or base change can affect the virus's function. So on the outside here is a diagram of the viral genome. It's an RNA genome, which means that unlike humans and a lot of other organisms, it's encoded only by a transcript, an RNA transcript. It doesn't have any DNA. It never goes through any DNA intermediate. It's one of the really fascinating parts of biology that these kinds of things can exist. On the one hand, that makes it a little easier to study. On the one hand, it makes it a little more difficult to study. But suffice it to say that RNA encodes for these different proteins. And a diagram of the virus is on the inside with its genome packaged inside a layer of lipids and proteins. And this red spike on the outside, which gives the coronavirus its name, or crown-like, is what is the primary thing that interacts with the immune system. And changes in the spike protein can affect both the antibody response, vaccine response, and infectivity, as well as the spike is used for interaction with human cells to create infections. And so it's a really, really, really important part of how the virus interacts with the world, and a place where mutations have particular impact. So as I mentioned, the standard COVID test or qPCR test does look at 3 bits of the genome, but it provides only a yes or no answer. And by doing genome sequencing using a set of amplification primers defined by a consortium that's used now throughout the world, the Arctic Consortium, we can decode essentially the entire approximately 3,000 base genome. And that gives us that detailed information to understand how strains are related to each other, how they've evolved from a common ancestor, and which mutations are showing up over and over again, and therefore are likely to be functionally important, and which ones are spreading more readily. And again, because it's a small genome, even small changes can be important. And many times, we'll sequence genomes that are perfectly identical to one another. The mutation doesn't happen all the time. And so when it does, it's something that's really important to look into. So just as one example, the B117 lineage is the UK lineage that has been getting a ton of attention, rightly so. This is a diagram of the spike protein. It's actually 3 copies that intertwine with one another to form a single structure. It's called a trimer, for having 3 equivalent parts. And so you see the same mutation listed in a couple of different places. But just to point out a couple of those, these are amino acid substitutions in the protein. And in this case, asparagine 501 is changed to tyrosine. And that single amino acid makes it possible for the virus to bind more tightly to human cells, and therefore more likely to get inside and cause an infection. P681H can also help cells become infected more, and other mutations affect the antibody binding. So knowing these mutations is important, both in understanding how the virus works and spreads, but also in tracking what's happening throughout the community. And it's that sequencing that gives us that fine level of resolution. So several variants have been classified, or the CDC has developed a variant classification scheme. And this is the full text. If you're interested in seeing and studying more details, I provided the link here. But there's now 3 sort of levels of variants that have been defined. Variants of high consequence, of which there aren't any yet, where would be direct evidence that there is an important medical response that would need to be provided that's different about a patient who is infected with a variant of high consequence compared to all the others. And so the fact that none of these have been defined really means that the variants right now, there is not a major change in how patients are treated or in how contact tracing is performed, et cetera. They're obviously of concern or of interest because of more transmissibility or something else, but there isn't a clinical feature that makes them treated differently. And the difference between a variant of concern and a variant of interest really has to do with the strength of evidence. So variants of concern, there is direct evidence of this increase in transmissibility or other features that make it concerning. Whereas variant of interest has some of the same mutations as a variant of concern, but there is less evidence to directly support these viruses as being, as carrying those health consequences. So we think of them more, we track both of them. It's certainly easy to keep track of the information, and it's very useful to understand what's going on with both variants of concern and variants of interest. And because they share a lot of the same important genetic markers, that's a reason to pay attention to both of them. So let's add some annotations here. B117 is the UK lineage. B1.351 is from South Africa. P.1 is from Brazil, and so on. B1.526 is from New York and 525. The B1.526 and B1.1.7 are the ones that are most common in Connecticut now, and Nate's going to walk through that in just a minute, showing you what's going on in the state in good detail. Again, showing some of these mutations. Some of these have been characterized both with a lot of in vitro experiments and cell culture, but also in recovered patients, trying to understand these effects on the disease course. And I would say that, first of all, for all of these variants, the United Kingdom, South Africa, Brazil, and others, the increased transmission, more efficient transmission, is now strongly supported based on data, epidemiological data, as well as other types. There is some evidence for some variants and in some contexts that there are also effects in other areas, including more severe disease, for example, with younger people, reduced vaccine efficacy for some vaccines for the South Africa and Brazil lineages, and in particular, reduced effectiveness of treatments. And by this, I mean the monoclonal antibody therapies from Lilly, Regeneron, and others that are often used in the hospital or for more severe cases. Certain of the variants have, as you can see on the right, much reduced inactivation by the monoclonal antibodies. So a few monoclonal antibodies have been had their emergency use authorization withdrawn from the FDA because of this, but there are currently several different options and some combinations that are quite effective. So I don't want to overstate the impact of the variants on monoclonal antibody therapy, just to say that this is an area that's of real considerable interest. The other area of considerable interest is in vaccine breakthrough, or people who become infected despite having been vaccinated. So first of all, the vaccines are incredibly effective, but they're not perfect. And so there's every expectation to believe that some people who have been vaccinated will in fact get infected. But there's a ton of research going on now about whether particular variants are more likely to infect people who have been vaccinated. It certainly represents a new selective pressure on the virus to have vaccinated hosts, but I think the jury at this point is really still out on whether or not, for example, B117 is more likely to be seen in patients who have been vaccinated. So where we are right now, to sum up, is a real race, I think, between vaccines that are protective and variants that are either spreading more rapidly or have the potential to infect vaccinated people. The data right now shows that, I think with the declining case count in the state, that vaccination is winning out. But it's something that's really important to keep an eye on going forward, is what the set of circulating strains are, and is that changing in response to the changing conditions here. So with that, I'll stop sharing and turn over to Nate to give the description of what's happening here in the state.
All right, thank you, Mark, for a great introduction to this subject. Hi, everyone. Thank you for having me here. I'm Nathan GruBaugh. I'm an assistant professor at the Yale School of Public Health. And for lack of other terms, I guess I'm a genomic epidemiologist. And that means I use genomics and sequencing, specifically of viruses, to track their emergence and spread. And before SARS-CoV-2, I investigated the Zika epidemic in the Americas. I worked on the Ebola outbreaks in West Africa. I do a lot of other work in our backyard of investigating West Nile virus and Powassan virus, which is transmitted by ticks, and a lot of other things. And when SARS-CoV-2 emerged early in 2020 in the United States, my lab completely shifted gears and focused on this. And now I'm going to take you through a little bit of a journey of what is going on in the state of Connecticut in terms of our sequencing capacities and also the variants. So to start, I'm gonna talk about sequencing capacity in Connecticut. I'm gonna walk you through this figure. These yellow bars represent the weekly number of cases in Connecticut through 2020. And so here's the axis for that. And then these purple lines represent the percent of cases sequenced per week in the state. And in 2020, most of this work was actually done by my lab at Yale. We're a small lab that does a lot of outbreak investigations and partnering with the Connecticut Department of Public Health on this. At the beginning of the pandemic, there was a lot of interest in sequencing SARS-CoV-2 to understand how it spread and where it was coming from, and we did a lot of work then. And then these little spikes that you see throughout is us working with the DPH on specific outbreaks. And here in August was our work on investigating the outbreak that happened in Danbury. But you can see that the baseline is pretty low. This wasn't a routine surveillance system that we had in place. So during this period in December 2020, when news of new variants were starting to spread, both the news and the variants themselves, we were in a situation here in Connecticut where we didn't really have a strong surveillance system to stand on to really know what was happening. So enter in early 2021, this collaboration between Jackson Laboratories and Yale to actually establish a routine surveillance system. And this was done with our own funds and on a voluntary basis. We decided that we wanted to do this because it was the right thing to do to help. And you can see right away that the percentage of cases had started to increase a lot. Also, it's easier to sequence a higher percentage of cases when the cases are relatively low. And the dip you see in recent weeks is not because our efforts are decreasing, it's only because it takes a couple of weeks for diagnostic samples to come to our labs to be sequenced and then that data to become available. So we always see a dip right at the most recent times. But where we want to be is at least sequencing 5 to 10% of the cases every week across the state. And that is to help us to detect emergent variants when they were very low frequency, when they're less than 1%, so that we don't get caught off guard. And you can see that our system has brought us into that, but there's still a lot of work to do to maintain this through the duration of when it is needed, and for as long as SARS-CoV-2 is around in the state. But our sequencing in the state is not uniformly distributed. The circles here represent the relative number of cases sequenced in the state, and then per county. And then you see the percentage of cases that had been sequenced as an average per week since the start of March. And so what we are doing at Yale and what Jackson Labs are doing is really sequencing by convenience. So we are, Jackson Labs has their own clinical diagnostic capabilities. So do we at Yale. I partner with the hospital. And so where most of these cases, where most of these samples that we are getting are coming from along the coast. And you can see New Haven County has a strong representation. We're near really that 10% mark that we want to have, but we have very little information in other counties. And so as this program continues to grow, which we're building upon this every week and working with the Department of Public Health, this is really where we need to have a refocused effort on surveillance. And that really means having the state level intervening and helping to get samples from those areas into labs that can do sequencing, which is actually not a trivial thing to do and certainly needs attention. So on to the data. So here I'm just listing, as Mark showed on a slide before, all the variants of concern and the variants of interest that have been identified by the CDC. I've put the lineage name here, plus where they were first detected for reference. And what you can see is that, and oh, sorry to finish taking you through this. So this column here represents the total sequence and reported to the state to date. And then the percentage of each of these that were sequenced here, I'm showing from the week of 4/11, because that's the last complete data that I have right now. And then the change from the previous week. And now I've bolded the lineages that are the most common in Connecticut, and that is B117, which is over 60% now of the cases sequenced, and then these two sort of flavors of B1526, which were first detected in New York, and together they make up about 20%. And the rest of them are pretty low frequency. So partially good news here. So B1351 from South Africa and P1 from Brazil, which actually has, which are most concerning for vaccines, are pretty low percent in our state so far. This here shows the weekly trends of the different variants in Connecticut since we started this program. And I have this extending back to December. And as you can see in late 2020 and early 2021, most of what we had sequenced, most of what we see in Connecticut were what we're calling these other variants. So things that are not classified as of concern or of interest. And you can see that this started to quickly change throughout this year. And now there's very few of these things that are not interest or of concern. And B117 in particular has been progressively taking over. And in the United Kingdom, B117 is now over 90% of the cases. And I would expect that to continue to happen here as well, where B117 just becomes the dominant virus that is circulating. Alongside of it has been B1526. And this has maintained somewhere in between like 15 and 20% frequency for a while. I have the suspicion that this will start to shrink out a bit as B117 continues its course of dominance. We can also look at this spatially, and this shows the data per county. And again, obviously some counties don't have a whole lot of data, as I mentioned before. And these are the frequencies of the different variants shown as a pie chart since March. And so, and B117 for all the counties is in red, and B1526 from New York is in blue. And you see here that in each of the counties where we have data, that the majority of the variants that are sequenced are one of these two. And perhaps in New Haven County, we have the highest percentage of B117, but that also may just be to having better surveillance here. But that is just to say that these variants aren't just important along the coastline. These variants are important everywhere, and we are detecting them everywhere. In addition to tracking the frequencies of the variants themselves, and as Mark has pointed out before, that we're also interested in specific mutations. The CDC has listed 2 mutations or substitutions of therapeutic concern. Mark has already mentioned this one that is at site 484. And if it has a K amino acid at this allele, this is a concern for vaccines, though it doesn't necessarily mean that all vaccines won't work for it, but it's of high interest that is being investigated. And this other one here at site 452, if it has an R amino acid at this position, it is a concern for monoclonal antibodies. And this mutation is primarily found in the variants that had been first detected in California. So you can see, and for both of these, sort of good news here is that neither one of these are like growing in frequency at this moment, and they're both low frequency overall. But this may change as the landscape changes with more people becoming vaccinated, and it's really important to keep on monitoring this. We, in addition to just looking at these, the frequencies of these over time, we also look for the detection of mutations that occur in some unexpected places. So this is a phylogenetic tree it shows the relationship of sequence cases. So each dot is a case, and its further progression to this side of the screen means that it has, that case has, that virus has acquired more mutations. And this is a representation of the B117 lineage that doesn't normally have this 484K mutation that we're worried about with vaccines. But in Connecticut, and as well as in many other states, we've found that this mutation is independently sort of emerged within this lineage, which is of high interest. But thankfully, because we are doing a lot of the sequencing, we're not seeing the rapid spread of this mutation within this lineage, but is one for us to keep an eye on. So why should we keep doing this work in Connecticut? Well, for one, we need to continue to track and detect new variants and mutations as they emerge, and we need to be doing a better job of it. We need to be capturing more areas, and we need more partners within the state to do this. As Mark has already mentioned, that we really need to be studying the impacts of variants on vaccine efficacy. Here I'm showing you some preliminary data from a study that we are doing at Yale New Haven Hospital, where we have sequenced over 1,100 cases of individuals that are not vaccinated from the same time period as that we've sequenced 40 cases of individuals that have become vaccinated. And the take-home message of this, with this preliminary analysis of what we find in the vaccinated population, and these are mostly the mRNA vaccines, so either Moderna or Pfizer, matches what we find in the non-vaccinated individuals. So we're not seeing an enrichment of one thing that might suggest that this is leading to breakthroughs. So this, to me, suggests that the vaccine breakthroughs are more likely to be caused by a host response. So not developing enough antibodies or not developing the right antibodies in response to the vaccine, and not specifically due to any specific variant themselves. We also need to do this work to continue helping the Department of Public Health with outbreak investigations, and more broadly to understand the patterns of SARS-CoV-2 spread within the region. Finally, I just want to leave you with this. You can find all of this information on our website, CovidTrackerCT.com, and you can follow us on Twitter at CovidCT. I will put this link in the chat in a moment. And on Thursdays at about 4:10 p.m., so later today, we will post a new update. We do this every week. It's at 4:10 because the state, the DPH, they release their data at 4 o'clock, and we'd like them to do it first. We work with them to make sure what we are showing matches what they are showing. We're all on the same team here. And this is a great way to follow our progress. And with that, I will stop.
Thank you so much, Dr. GruBaugh and Dr. Adams. Truly enlightening. I will say I particularly appreciated the Marvel slide. I'm a huge Marvel fan, and so I love thinking of it in terms of that. And I did just want to mention, I saw that Representative Steinberg has joined us. I'm not sure if he can be upgraded to a panelist or if he wants to say a few words, but glad to see Representative Steinberg was able to make it after all. And I know that there are some questions, and Lisa Roy of Jackson Labs will be going ahead and moderating those questions. Can I just kick it off with a quick question of my own? I'm just wondering if you could just basically tell us, you mentioned, Dr. Adams, at the beginning of your presentation, that it was, you know, a bit of a surprise to see variants popping up early on. I just wondered if you could tell us how typical this is of a virus to see so many variants. Glad to see that there are none of high consequence, because I would tend to think that that is not a good thing. But wondering how typical this is, that there's so many variants of concern, and also how they're reacting to the vaccine. I know you mentioned that 2 in particular have not been, you know, have been particularly resistant. And I'm just wondering what we're seeing out there in terms of that.
Right. So first of all, mutations in viruses is very common. And in fact, viruses have a pretty high mutation rate because their repair machinery, their genome repair machinery, isn't as well developed as it is in more complicated organisms. And in fact, the mutation and variation is part of their survival strategy as a way to be able to infect more hosts or respond to the environment. So mutation is a common feature in viruses. And a good way to think about that is in influenza virus, there is quite a bit of mutation to such a great extent that it means there needs to be a new formulation of the vaccine every year, and we need to get vaccinated with against the new variants that show up every year. I think it was initially expected or hypothesized that SARS-CoV-2 might mutate more slowly than other viruses. And that may or may not be the case, but the fact is that there are, nobody had ever been infected with SARS-CoV-2. And so there was a huge new population that was available. And the number of infections, the number of viral replication cycles has given a tremendous opportunity to experiment with new mutations. So the mutations happen. There's no directed, the virus doesn't know what it's doing by creating mutations. It just makes mistakes when it's making a copy of the genome. And every once in a while, every very rare event, one of those may have some advantage in terms of being more transmissible or giving the virus some other advantage. And then that can spread. And that's where we see those evolving more or arising in frequency. I don't know if there might have been more parts of your question, but I didn't answer.
No, that's great. And in terms of, vaccine efficacy, are we seeing, I'm just curious what you're seeing out there and what the expectation is in terms of how we're going to really fight this thing as these new variants pop up.
Right. So what Nate showed at the end, I think, is encouraging. Is that so far, and we've seen a little. I expect over the next month or so, 2 months, 3 months, there are going to be many more studies that look at vaccine breakthrough cases, as they're called. So people who have been fully vaccinated and yet nonetheless get infected. And it's going to be very important and interesting to track those, do genome sequencing on those cases to see whether or not they look like the general population. And of course, the general population is also changing in terms of their composition of variants. So time matching those is very important. But if they continue to look like what's circulating among unvaccinated people, that's great. It is true that the vaccinated individuals creates a somewhat different selective pressure on the virus. And so it's important, I think, to continue to watch this.
Okay, I appreciate that. And I promise, Lisa, I'll turn it over to you, but I do see, I just wanted to give Representative Steinberg a chance to say hi to everybody and our colleagues here. And I also see Representative Yaccarino with a question, and I know several of our colleagues have typed in questions. So I'll turn it to Representative Steinberg, and then we'll go to you, Lisa, to sort of moderate all these questions. Uh-oh, can't hear you, Representative. Oh boy. Representative Steinberg, you sound like you just took in a bunch of helium. And I think he's dropped off. Okay, we'll give him another opportunity once he is able to reconnect. And Lisa, I'll turn it over to you, and you can, it looks like he's connecting again.
Can you hear me now?
Yes, perfect.
Much better.
Yes, much better.
I seem to have this problem quite frequently, and I have to examine, or me, one or the other. I want to apologize for being late. This has been great. My excuse was that I was very focused on an article in the New York Times this morning, which seems very apropos to today's conversation, focusing on the B.1.617 variant in India, which apparently has had impact on those already vaccinated, particularly healthcare workers, even babies. And obviously India's situation is extreme and very distressing, but it seems almost inevitable that someone from India would get to the United States and eventually Connecticut. And this is another variant, which conceivably would be very concerning for those, as you mentioned, the whole issue of vaccine breakthrough or virus breakthrough. And, how do you account for variants popping up like this that, I mean, you're tracking them, obviously. How do you understand the risk that they pose?
Yeah. Thank you for that. I'll start with this, Mark, and if you want to chime in, please do. So yes, obviously the situation unfolding in India is just sort of horrible to watch, and there's a lot of things that I think are contributing to that, and I don't want to dive into all of those. I'll just speak to the variants themselves. So there's two specific variants in India. There's the B.1.617 and another one called 618, but and they do seem to be rising alongside of rising cases, but it's hard to say whether or not the variants themselves are actually causing that, right? We have now B.1.1.7 that is over 60% frequency in Connecticut, and we're able to drop cases. And we're able to still work on doing that before, vaccine rollout really was where it is now. So we don't have a whole lot of information about the true impact that this variant may have on vaccines. I think it's a lot of anecdotal evidence that's coming out of India right now. It's really important for us to study this. A lot of people are. We have not detected it in Connecticut yet. But one thing that's going to limit its opportunities in Connecticut is actually the fact that B.1.1.7 is dominant. I think this is actually a similar situation for B.1.351, which was first detected in South Africa, and P.1 that was first detected in Brazil, that we don't detect very many of these in Connecticut. And I think they might have not as many opportunities to spread when its competition is already one of, as we know of, the variant that is the most transmissible. So I think that it's going to be really hard to predict the future here and what's going to happen, but this is just why we all need to sort of work together, not just in Connecticut, across the US, across the globe on surveillance, helping each other out. And it shows that when you have, these situations like this that are happening in India, how they can actually have a potential direct impact on the United States, that we're all in this together. So I don't think I probably sort of hand waved your question there, and that's primarily because we don't really know yet, and I don't want to try to predict the future here.
Thank you, Dr. GruBaugh, and thank you, Representative, for that question. I see Representative Yaccarino has a question, and so we're sort of exercising our prerogatives as co-chairs of the Bioscience Caucus here. And then, Lisa, if you want to, Representative Yaccarino poses his question to the good doctors here, maybe you'd take over right from there. Go ahead, Representative.
Thank you so much. Excuse me. Thank you for your presentation. It's really informative. Do you feel there's enough resources in Connecticut? And I have a two-part question, and also enough sequencing of positive samples for screening, because I think you need those samples, don't you, for the positive variants or virus to screen, to have more data?
I can take a first crack at that. Both Nate and I, Jackson, Yale, are working closely with the State Department of Public Health in trying to expand the amount of sequencing that's done. We communicate regularly with them. I think it's particularly important, and where the state can help is in directing samples from areas that aren't as intensively sequenced to the groups that have capacity for sequencing. We sequence. I see there's another question in the chat about where the samples come from. Nate can answer that for Yale. It's a somewhat more diverse set that he's working with. The samples that we're sequencing are primarily through what's tested at JAX, and that in turn is the major component is through a partnership with Griffin Hospital that is conducting screening for the state in long-term care facilities, nursing homes, assisted living, several other places. So it's primarily asymptomatic screening. And then we have a number of generally smaller hospitals that don't have their own testing programs that we also support. But I would say the majority of our cases were asymptomatic when they came in, whereas I think for Yale, it's a much more of a mix between symptomatic and asymptomatic. And yeah, so I think we are certainly working with the state to try to increase both the overall number and the representation.
Thank you.
And maybe just to quickly elaborate on that too. So we do have the capacity in the state to meet what we need to do. Between the work at Yale, there's a lot of different groups that are very interested in doing this, increasing capacity. with what Mark is leading at Jackson Laboratories, not represented here, but UConn as well is increasing their sequencing capacities. And then there's other national partners. The CDC is directly involved in sequencing. There's ways to do this into that map that I showed to get that all to 10%. The harder part right now is just getting those samples from those diverse diagnostic labs into the labs for sequencing, which is not a trivial thing to do, because you have to get resources to those labs to pull out those positives, people that are working, nonstop doing the testing, to get them resources and time to find what they need to get that to a lab and everything. And so I mainly work with the Yale New Haven Health System, but I also have a testing partner, Murphy Medical Associates, that's in Fairfield County, which gets me more. I work with sequencing Yale campus, and we do some ad hoc work for the state where they send us things. So this is the new frontier. Like, we are establishing the sort of baseline of what we could, and then it needs to be expanded out to capture everybody so that all of our counties are represented.
So we need to get as much resource as possible. And I'll end it there, but I also like to say he's on the call. Ben Krinsky is a high school friend of my son, Dave. So he's on the call with us today. Hello, Ben. I don't know where he is, but my son just texted me, Ben's on the call. Thank you so much. Thank you, Madam Chair.
Thank you. And Lisa, why don't you take it from here and moderate the rest of the Q&A for us? Thank you.
Absolutely. Thank you very much. First of all, Dr. Pettit had a question regarding what you've already discussed regarding India. So Dr. Pettit, if that full question wasn't answered, please do enter a new one. But I think it covered most of what you were looking for as well. So moving on, this is a question for Dr. GruBaugh. Would you elaborate on the reasons people might not be developing enough antibodies to fight off the infection?
So I'm not an immunologist, so I don't want to dive too far into waters that aren't my expertise. But there's a lot of, we're not equal, right? We have different immune responses. We have different sort of host factors, genetic factors that might mean that you develop, and it's not just antibodies, it's T cells and it's other things too, that of whether or not you develop a robust response to the vaccine or not. Thankfully, this happens quite rarely so far, at least with the mRNA vaccines, that you would develop a response that would not be protective. And then there could be just some bad luck in it. It's hard to know all of that. I think it's sort of an evolving science, and I will sort of end that question there before I get too far down into something that I can't answer.
Great. So the next question is, do breakthrough cases tend to be milder?
I don't know if Mark, you have anything to add to that. There's, anecdotally, there's some evidence around this. There's still a lot of these studies that we're trying to do. I will just say from my end of things, sequencing vaccine breakthrough cases are harder because there's not as much virus there in a lot of these cases. But we need larger data sets to really get at this. But from the clinical trials from the vaccine, I will say this, that there were infections that had been noted, but there were no, from the clinical trials, they were 100% effective against severe disease and death. So in that sense, yes, the infections are milder. But now that we have a much larger trial of all of us getting the vaccine, I'm sure that there still are a few within that. So the general answer would be yes, they are milder.
Okay, another one, are home tests impacting your collection efforts?
That's a really interesting question. I believe, so there is antigen testing that has been happening in the state with Abbott Binax Now tests and others. And those results are reported in aggregate for the total testing in the state. I don't believe their antigen testing is split out compared to qPCR tests into the total numbers. The home tests are just now starting to be available over the counter and without going through a physician to order that. And that means that there is going to be the potential for a gap in reporting of positives if they're done at home. I'm not quite sure how those work. I understand there is an app for reporting, so it's possible the app will report a positive to the state Department of Public Health. I think this is a question I would like to know the answer to now that I've thought of it. Certainly the expanding testing, including home testing, antigen testing, and other ways, I do think is an important component of the overall fight against COVID-19, is that recognizing infections early and being able to take appropriate measures is good, and not the turnaround time for qPCR, although now quite good, ours is less than 24 hours from collection to result for the most part, is that turnaround time is important. It's not going to be 15 minutes like an antigen test. And while they're slightly less sensitive, it's still going to be an important part of routine surveillance going forward, particularly as cases continue to decline. Having that early warning system is great in areas where there are particularly congregate settings.
Great. So I'm mindful of the time here. I'm not sure if this will be the last question, but it's probably going to be the next to last. If anybody has additional questions that don't get answered, please do feel free to either enter them in the chat or email us directly, I'm sure, or the committee, Bioscience Caucus, directly, and they'll get those questions to us to be answered. So the question is, are the variants in Connecticut affecting particular demographics, or are they widespread from what you're seeing from your numbers?
So it's hard to us to get into fine resolution of demographics because our work as doing public health surveillance doesn't really allow us to get that far into details on some of this, but the Department of Public Health may have more numbers here because they have more information about each of these cases. But in general, we find that these variants we find them in all counties. So that already tells us that they're widespread. There's always been inequities of SARS-CoV-2 transmission. So there's likely probably inequities with these variants of affecting specific populations, especially our Black and brown populations in lower income areas. While that's speculation based on this without having the evidence for it, but that's based on what we have seen before in just cases. But yes, these are widespread and potentially important for everybody that lives in Connecticut.
And perhaps this will be the last question, since the second part of the other question. As more of the population is vaccinated, should we see a reduction of the variants?
I think that's a really good question. I'd be interested to get Nate's take on this as well. I think there's sort of, it's almost like multiple parallel pandemics proceeding in parallel. So the B117 is increasing as a proportion of cases, even while the total number of cases is declining. So they're like competing trajectories. The B117s are going up, but the total cases are going down. So which of those is going to win? And I think we're all optimistic that the declining cases will continue, and that's but the reason to keep an eye on not just the positivity rate is to understand whether or not, whether it's B117 or some other new variant coming up, is an early warning sign that the ground has shifted around what the assumptions are. And I think it's this genomic surveillance that can really provide that early warning that something different is happening than what's been happening in the past.
Nate, anything to add or any final thoughts before we close?
Maybe I was sort of typing some answers to questions to try to get some stuff out there, but maybe one that I would just like to say out loud to one of the questions about are we working with other states on this, or is this, and how does this, what we do in Connecticut, how does this compare? So Mark and I are involved in a sort of an informal Northeast research group. Then we meet every Friday, and it includes departments of public health from across the Northeast in many different institutions. And there we share information every week. We talk about what is happening in different states. We talk about the research. We talk about vaccine breakthroughs. And I think it's great for us then to get the insights from DPH officials in various settings, and for them to learn about what we are doing. And we know that Connecticut is not in its own bubble, right? What happens in New York affects Connecticut. What happens in Massachusetts affects Connecticut. What happens here affects those other places. So we try as much as possible to keep that information flowing. And then we also work directly with the CDC. They have a great program called Spheres that connects groups from across the country to do very similar things. And so there's a lot of information flow going on here. And but I do have to say that we're often sort of applauded for the efforts that we are doing here. The information that we are sharing is some of the best that you'll find in any state.
So we have one more fairly technical question. So this is obviously somebody who knows some science here. So I'm going to try to eloquently put this forward. If you think in the 5 minutes left, we need to answer this offline, please respond. But the question is, does the homology of the virus sequence to sequence of human proteins of known function offer any clue as to the course of the disease or to sequences of previously studied viruses?
I guess I'll start with this. This is a super interesting and loaded question that come to my class and we can talk about this for an hour. There's a lot of work on trying to predict how a virus might behave within a human based on the structure of the virus proteins or sequences and how they compare to the host proteins. I think they're good hypotheses to generate in these situations, but they're really, you need the clinical and epidemiological data to really sort these things out. There's a lot of experiments that are then done to see how viruses can evolve to best match host proteins and to figure out what mutations and therefore might be of interest. But none of that work, they're informative, but we need the real world clinical epidemiological data to really understand.
That is it then from our Q&A. Any closing thoughts from the Bioscience Caucus chairs?
Go ahead, Representative. I see you're off mute. Yacarino.
No, you go first, Madam Chair.
I was just going to say, I'm so grateful for this today, and as troubling as all of this really is and frightening, it's also incredibly comforting to know that we have, scientists on the front line of this really, working to understand what's going on out there, the data, researching it, and coming up with solutions based on that data. So I'm so grateful for this JAX and Yale School of Public Health collaboration. I hope more folks join in so that you are able to, in fact, get more samples across the state. And just want to say that as one of the Bioscience Caucus chairs, incredibly grateful for your work and here to help however we can further your research. So with that, I'll turn it over to Representative Yacarino.
Thank you so much. Really, I want to echo the words of Senator Cohn. And thank you, both Yale and JAX Labs, and all the scientists that participate, that make our world a better place. And, we need science, obviously. But thank you. When we need, not just state, but federally, not to give you enough resources you need for sampling, for testing, for the variants and other parts of the virus. But thank you so much. So. Well, thank you, Lisa.
If I may, thank you to the presentation and all the people who have watched and listened to this program. I think the key is raising awareness and education. Obviously, there's still significant room to go in regards to education and awareness. I would encourage that. Thank you for updating us on the fast changing dynamics. Ultimately, I think I leave this program with such tremendous gratitude and pride that we have such wonderful and talented scientists working so hard in the state of Connecticut. We're truly lucky, and at the same time, I'm truly grateful as well. So thank you very much for sharing your thoughts and insights with us.
And I would just like to say how much I always enjoy the science and the great work that is done at Yale and at Jackson Lab. And if anybody has any doubt, the key takeaway from this entire presentation is get caught up on your vaccinations. Thank you.
Hey,
Thanks, everybody. Be well and yay for science.